thank you so much for joining us at PWSA this evening to introduce ARD 101, the HERO study, to our community. We are extremely excited this evening to have Tien Lee and Muncie Jamin from Aardvark to walk our community through the ARD 101 HERO trial. And with that, I think I'm just going to go ahead and turn it over. Tien, I think you can maybe introduce yourself and Muncie, and you all are live.
Okay, great. Thank you so much, Dorothy. I really appreciate the opportunity to talk about Aardvark and ARD 101 and what we're contemplating on executing. We're really excited about it. So I'm Tien Lee. I'm the founder CEO of Aardvark Therapeutics. I'm a medical doctor by training. I've been a founder or co-founder for several biotech companies previously, and established Aardvark in 2017 with my wife, Jane, and Monty.
Thanks, Tien, and thanks, Dorothea. Thank you, everyone, for having us today. Really excited to share more about our company as well as this HERO clinical trial. I'm Monty Jamin. I'm a pediatric endocrinologist. I'm also the chief medical officer here at Aardvark Therapeutics. I always say that my North Star has always been the patients that I was able and lucky enough to treat. And so it brought me to Aardvark to continue that journey to bring life-changing medications. And so really happy to be here, really happy to talk about this and appreciate your support.
Okay, great. So I think everybody can see the slides, and I'll just advance through them and give a quick overview of what the company is doing. ARD 101 is the name of the investigational drug, and the trial is the HERO trial that stands for Hunger Elimination or Reduction Objective. And this is the phase 3 trial for Prader-Willi. And here's like our obligatory disclaimer, because we are a publicly traded company as of February of this year. And here's the centerpiece of our story, is really the dichotomy between appetite and hunger. And a lot of folks would, conceptualize appetite and hunger as, aren't they really just different extremes along the same spectrum of the same experience? And really, they can be defined as different neurologic drives for your brain. They both govern how much you eat. One is a pleasure-seeking drive, which is what we think appetite is defined by. And one is a pain avoidance drive, which is hunger. So you can think of that as like a carrot and stick type analogy. And when you're really have an appetite for something, a food that might be very delicious to you, whatever your favorite food is, whether it be ice cream or carrot cake or whatever it is, it's appetite compelling you to eat that thing. We think that the GLP-1 drugs, a lot of the obesity drugs, are primarily anti-appetite drugs. They diminish the pleasure you get from the food. There's a sense of nausea that is related to the dose effect of the drug, and it basically turns away the reward of it. At the same time, you have hunger, and hunger doesn't really manifest for normal individuals with that much regularity. But the way to kind of describe it in the experience is that if that ice cream food that we mentioned earlier, that turned into dog food, for instance, and no one would argue that the appetite would go to zero, and most people still wouldn't eat that dog food. But if there was left alone to just only having dog food over enough hours or days, eventually it would start to look like you have to eat it, and you wouldn't enjoy the experience, but you're just trying to get rid of that pain of the hunger. And both these are managed at the same time in your brain, and the normal trigger to alleviate both appetite and hunger is food. When you eat food, your body releases GLP-1 to satisfy appetite, and it releases CCK to satisfy hunger. And normally that's the feedback loop. You get hungry, you have appetite, you eat, and then it basically feeds back on that. That's the normal circumstance. And we think in the experience of in Prader-Willi, it's mostly a hunger-driven process, not an appetite-driven process. It explains why drugs that are normally effective in general obesity have not really shown to be as effective in Prader-Willi, because we're taking away the reward, but that's really not the driver of the condition. Our drug, ARD 101, is a drug that activates the anti-hunger portion of it through that CCK signaling. So it has some unique properties. Our drug is an oral drug that you take by mouth, and it's hitting actually bitter taste receptors. And a lot of folks would think about bitter taste receptors as far as conveying taste in the mouth, and that's exactly what it normally would do. But they're actually bitter taste receptors all along your gut as well. And our goal with our drug is not to trigger the bitter taste receptors in your mouth, because we don't want anyone to taste our drug. It's very bitter if you were to taste it, but it's shielded. So you take it by mouth, and the contents are released in the GI tract, and it's activating bitter taste receptors in your gut. And our drug is 99% gut restricted, which means very little of the drug gets into your bloodstream or the rest of your body. Normally, that's not a feature you're proud of for a drug, because your general intent is to have a drug go where you need it to be. But in this case, the target of our drug actually resides in the gut. So we're triggering bitter taste receptors in the gut, and those bitter taste receptors in turn make the intestinal cells release CCK, which then activate a nervous system signal to the brain to shut down hunger selectively. So that's how we're getting at hunger, through this gut-brain axis. So it's actually working like a domino, first through bitter taste receptors, then to release CCK, and then to turn off hunger. So I just want to frame that discussion to see how we're differentiated, and why we think that our drug is very appropriate in the treatment of Prader-Willi syndrome. Now, when we actually were doing the research work with our drug, we were actually trying to get really into the science, and we were exploring the many possible uses for these bitter taste receptor drugs. It's been observed that bitter foods are generally related to eating less, and we wanted to find out why that was the case, and whether hitting just the bitter receptors outside your mouth, because you won't want the feeling of having eaten something extremely bitter, whether that was enough to turn off eating behavior. That's when we discovered it was actually working through gut-brain signaling, and then it was actually working through anti-hunger, not anti-appetite, and it makes it very unique in that manner. So then we're looking at the spectrum of different conditions, and what is the condition that best matches the drug. So we were picking the problem to match the solution, and frankly, we weren't, in the beginning, set out to treat Prader-Willi per se, but we looked at the mechanism in Prader-Willi syndrome and what was driving a lot of the clinical manifestations, and it really kept on bringing us back to hunger, not appetite. Now, I want to kind of step through the many layers of rationale why we thought this was appropriate. So there's actually some 3 big hints that give us an indication that something is more hunger than appetite. And number 1 is, if you decrease food quality, does that really dramatically change the overeating? And for folks with general obesity, I mean, if you decrease food quality, that would actually slow down because the food would become less rewarding. But as you know, in Prader-Willi syndrome, a lot of these individuals with Prader-Willi, they feel this incredible discomfort, and it doesn't really diminish their drive to eat, even if the food available isn't of high quality. Number 2, the second hint is, if GLP-1 drugs like the Ozempic and Wegovys of the world, if those haven't really shown to be as effective, maybe it's a hint that you're dealing more on the spectrum of hunger instead of appetite. And the third is a blood hormone. It's like the gut peptide hormone. It's ghrelin. It's like a kind of a blood marker that correlates with how hungry you are, right? If you have a ghrelin levels that go up and down throughout the day, if they're elevated, you feel like you're really missing a meal, and you feel kind of hangry. If you have a high ghrelin, even 20-30% above normal, you start feeling something is off. In the case of general obesity, it's really not that different than regular individuals, but in Prader-Willi syndrome, it's up to 3 times higher than normal. So it's a reflection of how much discomfort of hunger is being experienced. So they have 3 different hints that something is more hunger than appetite, and Prader-Willi sets all 3 of those criteria. On top of that, there's particular relevance for CCK as well too, because Tony Holland, one of the great investigators of Prader-Willi, and it really elucidated a lot of the science behind it. He noted over 30 years ago that Prader-Willi individuals, they actually can make CCK just fine. So your baseline levels of CCK, that hunger regulating hormone, the baseline level is not that dissimilar than normal individuals, but the normal trigger for releasing more of it to satisfy the hunger that you have is food. But when Prader-Willi individuals eat food, that CCK is not released in the manner that you would expect. So it doesn't really increase appropriately with the food. So that trigger somehow is broken. So although Prader-Willi individuals can make CCK, somehow they're not releasing an appropriate amount when they're eating food. So that trigger is broken. And we believe that our drug is a secondary trigger that can induce the release of that CCK we know that they can make, and that can give that feedback loop. But there are actually more hints than just that, because as we know in Prader-Willi, it's not just about the hyperphagia, though a lot of people might focus on that. It's definitely a very main a quality of life impediment. But beyond that, there's a lot of anxiety that's generally associated. There's inflammation, a lot of joint aches and pains, and these inflammatory markers that are elevated. Oftentimes, even if you control food access and a Prader-Willi individual is normal BMI, there's still a higher than normal body fat percentage. And then there's also that extreme constipation that seems very prevalent in Prader-Willi, where a lot of the drugs that you normally take to alleviate constipation just don't work as they should. And there's something slow about the way the gut processes food. So something that you eat in a meal would take, sometimes days to end up in number two. And all those things are clinical features in Prader-Willi that are common. And those are all processes that are governed and manifest by insufficient CCK. So low CCK, low gut-brain signaling leads to increased anxiety, this high body fat percentage, inflammation, as well as slow gut transit time. This even correlates with gallbladder contraction, and individuals with Prader-Willi syndrome often have gallstones at an earlier age too. So this all is, again, hinting that CCK is the issue, and have not enough of it is really what's driving a lot of the clinical features. And we're very, very hopeful that because of the way our drug works, that we're ultimately able to alleviate, or at least have a rationale why we can address all these clinical features. So as a summary of what work we've done to date, our company was established in 2017. We started working out the science. We started to work in animals, of course. We had a phase 1 in healthy human volunteers. We had 3 phase 2s, of which one of them was in Prader-Willi individuals, but we also had a phase 2 in general obesity patients and in post-bariatric surgery patients, just to understand our drug more. And in totality, we've dosed over 70 humans with our drug, and it's shown to be very well tolerated. It's the safety is really, we think, quite remarkable, especially as you benchmark it against other experimental drugs. We've never seen more than a grade 2 adverse event, and those are, on the spectrum of things. I wouldn't want to diminish anybody's adverse events, but there's grade 3 and grade 4. We didn't see any of those, those adverse events were, even if it's some itching in the skin and so forth, I mean, they're generally transient, resolved, and even patients that were dosing through, the majority of the adverse events that we noted were with continued dosing. We saw resolution with continued dosing, so that's very encouraging. The degree of benefit that we saw as measured by HQ-CT9, which is the approvable endpoint with the FDA, was actually quite substantial, we believe, especially in reference. We actually had a fairly low bar to be included in our phase 2 study. So the normal circumstances in HQ-CT score 13 or higher to be included. We only had 10 or higher to be the inclusion criteria. So as a result, our average starting HQ-CT was not as high as some other studies in the competitive space, but we saw really dramatic decreases. And I really want to underscore that this decrease was only within a period of 28 days, a very short amount of time. The reason why this study was a short amount of time is back then, when we conducted the study, the company could only dose for 28 days because we didn't do the requisite toxicity studies to allow for longer dosing because we were still bootstrapping things with a constricted budget in the early days, trying to get private financing for the company. We've now since completed the 9-month tox study, which should allow for indefinite dosing. So now we're at a point where we can dose for as long as needed. We completed dosing in 18 subjects with Prader-Willi syndrome. There was one patient that withdrew, but there was 18 patients that completed dosing. And now, inspired by the anecdotes of response and encouraging signals of effect, which include a lot of the suggestion of possible benefit beyond just the hyperphagia, we now have the HERO phase 3 study. And it is a worldwide study. I won't take away Monsi's thunder, but we have many sites that we anticipate opening for this. And the intent is for a 12-week endpoint, and I'll have more on that a little later. So as a summary of what we've done, in the first part, we had dosed a low dose of 200 milligrams twice a day for 28 days. So the first 12 subjects that completed dosing, you'll see on the left. The 28 days, you see this reduction of HQ-CT score. We actually saw for the patients that had HQ-CT 9, a score about -9 on the HQ-CT. And then that shaded area is that withdrawal period. Now, again, only 2 weeks off of drug this time. We had a very compressed study. So just within 2 weeks, we saw a trend of withdrawal, which is, again, suggesting that the drug, while it was given to subjects in the treatment period, was conveying a benefit. So that was very encouraging. On the right, you see data from the patients that had the higher dose. So we were exploring higher doses at 400, 600, 800 milligrams. So all these subjects got dosing at 400 milligrams for 1 week, and then 600 milligrams twice a day for 1 week, and then 800 milligrams twice a day for 2 weeks. Our go-forward dose for phase 3 is indeed the 800 milligrams BID dosing twice a day. We chose this dose because it's still very far away from the theoretic toxic limit of the drug, because the drug is, as I mentioned before, it's gut restricted. So it's primarily just stays within the confines of your gut. So we think that is very a significant reason why we think it will be safe and well tolerated in general. You see some of you have two graphs here. So the patients that stayed on protocol had the -7.8 at the second set of 6 patients, with a couple patients that just because of the way we were defining the parameters of the study, that they weren't complying to the protocol of the study, but we included that data anyway so you could see it. But we think even on balance is very, very suggestive of a consistent effect. And again, you see that withdrawal period that really shows a rebound of it when you're off the drug. So again, all very encouraging and supportive of our phase 3. And then when you look at the breakdown of responses that we saw on the left, you see the patients that we took as an overall group. They saw a -30% reduction of the HQ-C. So I want to emphasize that, because the study was very short, but despite that, we saw a pretty dramatic decrease. I mentioned before, the starting HQ-CT was not as high as other studies because of the inclusion criteria, but we saw a very substantial decrease. And then if you actually look at the patients that had a profile and the parameters that are similar to what we were expecting to reflect our phase 3 population, the average response of those groups that have similar setup had a -48%, again, in just 28 days. So we thought it's very, very encouraging. We're not trying to prognosticate what the effect is. This is a very small data set, of course, but we think that this is all very, very encouraging and suggestive that we should proceed with the phase 3. So I hope you agree that that's quite encouraging. And then as far as the study design for our phase 3, so there would be a screening and there would be a secondary screening period just to get a baseline assessment of the HQ-CT score. And then there's a there's two arms, so there's equal probability that one would be on either the treatment arm or the placebo. At the end of 12 weeks, that HQ-CT score is compared against that baseline measurement, and that is the benchmark by which we'll try to possibly seek regulatory approval on that HQ-CT score. But we're very, very encouraged that we'll have the likelihood of seeing positive signals for a number of other things that we're measuring. We're looking at PADQ to kind of have a proxy for anxiety. We're looking at DEXA scans to look at body composition. We actually shared data a quarter ago showing that there was a really strong trend of not just reduced body fat percentage in that 28 days, but lean body mass, not just preservation, but as a group net gain of lean body mass, which is actually really consistent with our expectation given our preclinical data. And also, we're looking at inflammatory biomarkers as well. So all the things I mentioned in terms of the possible utility of invoking this CCK response, we hope to see those clinical signals. We're looking at clinical global impression, and there's a lot of different things that we'll be measuring, but we're trying to make it very easy for patients, so we don't have fasting involved. With a lot of remote visits. We're trying to make it easy in terms of participation in the study. Again, not to take away Monsi's thunder, but we had a lot of these things in mind, and we had feedback from PWSA, of course, for how to optimize this as well. And the important part of this is that at the end of the study, there's an open label extension. So our intent is to offer open label extension to everyone who participates. So even if you're on the placebo arm or the active arm, there's an opportunity to continue to be on the drug after the end of the formal end of the study per se, because we don't want to have to take patients off the drug if they want to continue to be on it. So that was the reason why we are doing the full start of our trial now, because we wanted to wait to ensure we were able to offer this to everyone, and we wouldn't be able to do that unless we completed that 9-month talks, which we've now completed. So I'll leave it there. I think that's the last slide. That's right. So I want to make sure give Monty a chance to talk, and of course I'm sure you have lots of questions. Be more than happy to address any and all of them.
Thanks.
Monty, before you get started, I just want to let everyone know we've had a couple of people enter their questions into the Q&A, which is the live portion. The chat portion of the webinar tonight has been disabled. So if you do have a question, please go ahead and put it in the Q&A. I also have people who have been texting me questions, so we will get to that. But Monty, I'll let you go ahead and get started.
Oh, thank you. I don't have much more to add, except I always like to recap, because I think it's important to make sure you kind of hear the bigger picture items, because Tian is excellent at explaining the history and also kind of how we got where we are. I am a pediatrician to the core, and so when I explain things, it's to parents. And so I want to make sure that you understood kind of why we approached the phase 3 program the way we did. So a couple of things we mentioned were the hunger hormone. So we're looking at the hunger hormone that shows up when you're actually hungry, which is ghrelin. We're also looking at CCK, which is how we actually think our drug works in that pathway. CCK increases to give your brain feedback that, hey, you're full and you need to stop eating. And those are the kind of the seesaw effect that you should see. It's the positive and negative feedback loop that we're trying to fix. And specifically the feedback loop that tells you that you should not be eating anymore. That's the first piece. The second piece is that phase 2 design that we had for PWS specifically. What did we see? We saw that there were markers that it was tolerated. This drug specifically was tolerated, that at that study, we saw signals of safety that allowed us to move forward. Also, we saw the changes in hyperphagia and also the changes in anxiety, part of the assessments that we did anecdotally with the sites and the patients and their caregivers or their families. That was very encouraging. Because of that, we were able to move into this phase 3 design to really see if these signals are true, they last, and they last longer based on a longer duration of giving the medication itself. And so I wanted to make sure I circled back on that. And then looking at the specific things that Tian already mentioned, which was really important for us. coming as parents. Both of us are parents, but we also are doctors. And we've seen patients and we see the burden that you have just in your own disease process every single day, especially the families. So as we approach the phase 3 program, we wanted to keep it as low burden as possible while giving a chance for this drug to show what it can do. And what he mentioned was that we decreased the number of visits. So we actually only have 4 visits in the entire study that are in person. 3 are televisits, which means that you just pick up the phone, you call the provider, you talk through some of the questionnaires, and that's basically it. And it's to keep it minimal at those visits. Second is that we took out fasting. In a study that's designed to control hyperphagia, and specifically in this population, we wanted to decrease that extra burden that you can have in these clinical trials. And that was, again, very important to us. Everything we've done so far has been to ensure that patients are treated as partners and their families are treated as partners. And hopefully that's reflected as we execute this trial. I know there's a few questions that are in the Q&A, so maybe I'll pull that up as well, Dorothea, and answer some of them because I can see them.
Yes, that would be great if you can do that.
Okay, and so I was going to get to this open label extension, which was really important for us to give everybody who participated in the phase 3 a chance to actually receive active drug. And so that is something that we will implement, we're planning to. And then the question was about younger children. So 100%. We believe this drug should be accessed to all age groups. And so we are planning a study that will be for kids under 12 years of age. More to come on that, but I promise that is on the top of our list of things to accomplish. And then younger patients have better results. How young? So that was another question that was here. So in general, so the data is under 18 years of age. So when hyperphagia shows up, which is kind of at peak 7 to 8 years of age, though it shows up earlier, but it peaks with that behavior around that time and continues. And so these studies that show are within that age group that they have severe hyperphagia and when it actually shows up, and then under 18 years of age. So it's variable in between that time. But we're saying just younger than adult age groups. Sorry if I missed this, but how do you measure HQCT? So it's actually 9 questions. And a fun fact is that HQCT, it was actually called Hyperphagia Questionnaire by itself. CT was added for clinical trials. And so it was developed as an assessment tool with 13 questions for clinicians. So they'd see patients, they'd give the HQ score, and out of those 13 questions, and that I think that was 0 to 5, and the HQCT is actually 0 to 4. But either way, it was developed to assess how hyperphagia has changed for patients, for a clinician. And when it was time to start doing PWS studies, there was nothing that was validated that could be quantified for clinical trials for regulatory agencies. And so early days, when the first PWS trials were being done, they developed the HQCT9 based on the questionnaire that had 13. They removed a few questions because some questions were basically, at what age did you have hyperphagia? And you can tell that's a one-time question. It doesn't change over time. So questions like that were removed that weren't changing over time. And that's how they developed the HQCT9 specifically for clinical trials. So that's how HQCT, which is clinical trials, was developed. It's important to know that just so you understand kind of it wasn't just out of a random spot, but it was really developed through a questionnaire that was already in place for clinical assessment. And that is, so the question was, how is that executed, administered? So it's given at different time points throughout the trial to assess changes over time.
And you don't mind, I'm looking through the Q&A. I want to make sure I get everyone's question, and I don't want to miss anyone. Yeah, so remind me, Dorothy, if I missed anything. But yeah, so for right now, the HERO trial is eligible for patients 13 and up. The reason why, just so you know, our phase 2 was only ages 17 and up because it's a new first-in-class drug. FDA requires substantially more preclinical data to enable younger patient dosing. So now for phase 3, we are able to do 13 and up. We have full intent to try to aim for including patients as young as 5 years old. So we are actually working on that, and it's actually subject to a future announcement. So if you have a child that is below 13, all I can say officially is stay tuned. We absolutely have that in mind, and we're working on that, and we hope to make an update in terms of what we plan to do in terms of trial participation there. And then and I also just want to comment, younger patients tend to have greater responses. It's an observation that in the Prader-Willi clinical trial space, it's generally been a pattern of observation that regardless of the drug, whether they have been able to get approval or not, younger patients tend to be having more substantial responses to treatments, investigational or otherwise, and that's generally an observation. Just wanted to note that the data that you saw us present is based on patients 17 years and up, and we also saw general trend lines of younger patients having better responses even there, and we're very encouraged that as we look at younger patients in our study, we may even see, if we're similar to other experiences, even a greater responses. And again, the HQ-CT improvements that we saw, we think generally the other observation among the Prader-Willi trial space is that the longer you treat, the higher doses that you give, the younger the patients, the better the responses are expected to be. So we are actually having those parameters in favor of our phase 3. So that's why we're very encouraged. Of course, biology is difficult. I don't want to prognosticate what the outcome is, but we have reasons to be optimistic there. And then another question about to be eligible, participants have to have a single designated caregiver who can attend to get. Okay, so the reason why is there just needs to be a consistency of who fills out the HQ-CT. However, it doesn't require someone to be there 24/7 to observe the subject. I think there was is it a far minimum, or there's a certain window of time where that caretaker can observe. The reason why is because different individuals filling out the HQ-CT in referencing how a patient is doing, the scoring might be different depending on the individual rater. So we just have to have consistency, but it doesn't require a person that, if you have two working parents, it's still possible. It's just a matter of there's a minimum time designated for how much observation time you can have to have a consistent reporting. So I wouldn't, I want to defer to Monsi on that one too, for the
if you will, as a parent who has a child living with Prader-Willi syndrome, who's been participating in the clinical trial, I can tell you that the consistency of the person that is filling out the surveys and the reporting guidelines within these clinical trials is it really is very important. We all see hyperphagia and things that are going on with our loved ones who are living with PWS through different lenses, and being able to capture that primary caregiver's perspective really ends up helping to solidify what the data is going to look like for these clinical trials. So that's really, I think, why most of the clinical trials request that one person, the same caregiver, is filling out these surveys.
Yes, absolutely. And the caregiver requirement is about 4 waking hours per day on average for the week. And so we gave enough flexibility, but acknowledge that that's the minimum requirement for that caregiver role. And it does need to be consistent. We understand life happens, but I think that for that's why we minimize the number of visits. And even the televisits can be done through the phone. So that hopefully will allow for that same caregiver to have availability to fill these out.
Yeah, and I'm going to jump around. I want to make sure I get everyone here too. So yeah, no, not an age restriction for the parent age. That's not going to be the case. And then as far as the bitter taste, the real unifying purpose here is to shield the drug from being tasteable. It's really redundantly coated, so even if you were leaving it in your mouth for many minutes, you shouldn't actually accidentally taste it. Now, that's not to say that it's impossible, but there's a lot of layers to protect against it. I think we had a subject with a patient with a history of reflux that actually did note a bitter taste, but it wasn't so aversive. They were able to complete the dosing. So that was happening in the middle of the study, and they were more than happy to, from what I understood, to complete the dosing. So it wasn't something that was at least in that case prohibitive for continued dosing. The question about risperidone. Yeah. Okay. So we as a company feel very strongly that we would. Okay. Dorothy.
No,
Or,
no,
okay.
I was just saying you're answering it live.
Okay, great, great. Yes. Okay. So I thought you personally wanted. Okay, so never mind. I'm slow here. Is that so? We actually have really high optimism that even for patients with risperidone, this drug would still be of benefit. The reason why is a couple-fold. Risperidone is a medication that activates another part of the brain to increase appetite as a side effect of the drug. It's hitting this 5-HT2C receptor. I don't want to get too much into science, but we think it can potentially mask the benefit of our drug because the HQCT score doesn't really delineate appetite and hunger. So if you're introducing appetite in the context of someone with Prader-Willi with risperidone, the hunger is dominant. It's really the main issue, and that you really won't see that much of a effect of the risperidone. But if you're alleviating the hunger, but you still have an overlay of appetite because of the drug, it could kind of minimize the signal. We still think ultimately we're hopeful that patients that are on risperidone oftentimes are managing the behaviors that are relating to the hunger in the first place. So we're optimistic that even patients with that could potentially lower their dose or not even have to take it. But it's another factor. If you think of a clinical study, there's so many variables. It just makes the algebra equation a little more complicated to prove that our drug is working. So just for the purposes of our phase 3, we just are not including them. But we do have intention that we would be the drug would have ability to address those patients too. I just want to explain the reasoning behind that. Yeah, go ahead. Sorry.
Actually goes with the other question about why are type 1 diabetics excluded from the trial. And so what we did was you want to see a clear signal of effect, really, because that's the most informative piece to get this drug actually available to patients everywhere. And in order to see a clear signal, you have to take out the variables, as Tian mentioned, that can affect eating behavior, because the entire assessment is based on changes in that behavior. So if you're giving a drug that could potentially change it, it can alter the results. And so what we tried to do is simplify that and take out as many variables. Yes, we had to add some of the medications that some of the patients may be on, but we wanted to make sure that we have pure results that would inform efficacy when it is in fact available for everyone. And we truly understood that full effect. But if you have drugs, insulin can actually alter eating behavior. Risperidone and some other antipsychotics and medications can alter eating behavior. So we have to remove that from this trial, short trial, in order to truly assess the full effect, because we do want this available to everyone. We don't want these to be part of the caveats and how do you take the drug, but we won't learn enough about it if we have that in this clinical trial, which is why they were removed. And so that was one of the questions. And there's another question that's right behind it, actually, that I'll answer. Is this drug good for a 35-year-old? And so this should work on all ages, right? That's absolutely true. And actually in our phase 2 trial, we had mid-30s in the clinical trial as well. And so they participated, they continued, completed. And so, yes, it is good in terms of being available. And we'll see in phase 2 and phase 3 in a larger population how effective it is. We're very, very encouraged by what we've seen so far. And then what is the oldest age individual in the trial? I will say mid-30s for the phase 2, but remember it was an N of 18. So this is why phase 3 is so important, because we'll get a larger group of participants and more data just to inform me about what we are seeing already.
What is the cap age for your phase 3?
There isn't one.
There isn't one. Okay, perfect.
It just has to be greater than 13, greater or equal to 13.
Okay.
Yeah. And then I kind of think I'm going through these questions on the side. So yes, we are planning the younger age group.
Muncie, while you're reading through, I'm just gonna make something clear, because I do see a lot of questions in the chat, folks, where you're asking about ViCat XR, versus Aardvark's ARD 101. I want to be really careful about what it is that we're talking about here right now. So I don't know that that our folks at Aardvark are able to answer those specific questions. If you have a question or a concern as to which drug might be appropriate for your loved one living with PWS, that's really a conversation that you need to be having with your physician. And you know, that answer is going to be different for every single person. So I don't know if you've asked a question in our Q&A that relates to VYKAT versus ARD 101 or a VYKAT specific question. I don't think that that's something that we're going to be able to answer on this evening's webinar. And I would encourage you to get with your physician and ask those specific questions with your specialist.
I can actually try to attempt to answer some of that. I was actually about to make an attempt to do that. I want to be very careful too, as you say. You know, we respect the folks at Soleno, and you know, VYKAT is approved, so it's actually a benefit for the community. So I do want to address it, and not to diminish anything away from VYKAT, but I think it's a different approach that is you know, ultimately intrinsically not in competition, really. But you know, we think, I'll just answer it in the positive for us, right? I mean, we think that our drug's mechanism and the way it works is, in our view, directly relevant to really what drives a lot of the manifestations of Prader-Willi. And I want to emphasize that it is our sincere belief and hope that it's more than just hyperphagia that we could potentially address. That CCK signal loss is related to anxiety, inflammation, and that constipation, along with some other things too. And there's other evidence. Let me just share, just be a little science nerd and share you some reason why we think it's evident, right? If some people have abdominal surgeries and they have they cut part of their vagus nerve. So this is actually what our drug is working, we believe, is that we're stimulating bitter taste receptors that in turn like knock the domino of CCK, and that triggers this vagus nerve activation that goes to your brain stem to turn off hunger. If you have a snip in your vagus nerve through abdominal surgery, normal people will often get hungrier. They gain weight, increase body fat percentage. They also get constipation. So it's that signal that it's really tied to a lot of things. If you do a genetic experiment and in an animal knock out the CCK receptor, you have unending hunger as well too as a manifestation. So we think that the relevance of our drug is on target. We're also, I would just say, it's always apples and oranges when you look at clinical studies, because they weren't in the same study. But when we looked at our data, and of course there's a lot of caveats because of the small number, the signal strength, the suggestion of how strong our drug was very encouraging to us. And again, the anticipated safety profile, the lack of adverse events of a higher degree that we saw, it basically, we think we're very confident that it's a well-tolerated drug. So I'll just say in the positive, and of course the decision in terms of the differentiation with VYKAT, I'll leave up to your medical providers to delineate. But I just think that is a kind of a rationale for why we think that we'll be contributing something in the treatment paradigm. And of course, there's other exclusion criteria for VYKAT too. If you have renal insufficiency, that's an exclusion. But because our drug is gut restricted, it's not really challenging the kidneys in our mind, so that wouldn't be an exclusion for us. For instance, that's one category where patients that wouldn't be qualified for VYKAT would potentially qualify for our study, and so forth, right? So there are actually a number of reasons I could go on for longer than the allotted time on it, but of course, you know, I have a view that's Aardvark centric, so I just wanted to disclose that as well too.
And I would encourage everyone, really look at safety profiles for anything that you participate in, any clinical trials, not just ours, right? Look at what you can learn about safety profiles. You're asking the right questions. Ultimately, Dorothea's right. You have to talk to your own provider to see what matches what you need. And it's individually based. We are encouraged with what we're seeing. We're excited about the phase 3. remember the terms that Tian's used: gut mediated, gut restricted. It stays in there. And I love that he always uses this, and I don't think he used it today. Poop kinetics is what we're about. So you take it by mouth and you poop it out, and it has limited effects elsewhere. And that's what we're trying to continue to show in the phase 3. And I think that's going to be really important for everyone to understand. And as you approach clinical trials, these are the questions you always want to look at the back of your head. Safety: what have they shown previously? How does it actually affect the body? And then make the decisions based on those key factors. We're really, really excited that the space is now opening up, because this is a long time coming to have therapy to treat hyperphagia. Finally, you will have options, and we're hoping to be one of those that can truly give you meaningful change in your life and in your family's life. And again, thank you for participating in this, because we're learning a lot even through these questions on what's on the top of your mind as well. Because as we enroll for this trial, I'm sure these are the type of questions we'll probably get during our site visits to the PIs that are running these trials. And so I encourage you to ask this to your provider, ask it when you go to these clinical trials, make sure you're aware as you participate. That's also important. But again, like I said, we're very, very highly encouraged.
Folks, there is a question on here that I wanted to see, Muncie, if you and Tien might be able to address. And I think I know how I would answer it, but I think it might go a long way into helping to continue to educate our community about how clinical trials work and what the data looks like. And there's a question here that talks about the community's learned a lot about VYCAT from Soleno, representing a major advancement in recent years, and that their data is available to the public, and yours is not yet. And there's a question within this question which talks about which drug might be better. But I think from a data perspective, I think it's really helpful to make sure that community members understand the difference between how phase 2 data is published versus how phase 3 data is published, and what that looks like. And it's all publicly accessible. But is that something that you all can speak to really quickly?
Yes,
Okay.
absolutely. Tien, both of us maybe can interact on this one. But so phase 2 data. So there is some data available through abstracts. We haven't actually had a publication, but that is pending. We're hoping to have something that's a lengthier, all-encompassing one. But we have abstracts that are available through actually a lot of these meetings that we've been on and at since last year. So even presented some of the part 1 phase 2 data, and then we have part 2 phase 2 data that you'll see in less than a month. And we're hoping to share more from a publication standpoint, but also the phase 2 is important to know. The early data is out there, and we do actually share that through our the process that we're doing now with our website, which is the Hero for PWS. In case you haven't been on there, it's Hero for PWS. It's a plug only because a lot of the questions you may have about the trial are actually directly on there. So do the research, go into the website, see what you find, and it'll also give you some of the other questions on here are sites, global sites. What are the sites? What are what's the total number of participation for the size that we're having? So right now it's 90. It's a competitive. That means that any site that enrolls can enroll. We just want to give everyone the fair chance, but we want people to be ready to go into a study as soon as sites are available, and we will update that website when sites near you may be available to check that website, or clinicaltrials.gov. Now, in terms of the data availability, like I said, phase 2, it's a lot of these studies are on clinicaltrials.gov, right? So when you do these clinical trials, especially starting phase 1 to phase 2 to phase 3, you put in the specs, kind of the main points of what the clinical trial is about on clinicaltrials.gov. That's a requirement, especially US run clinical trials. You put them up. It also tells you usually the sites. It'll tell you the inclusion, exclusion criteria, the main ones, and then kind of when it's recruiting, when it's completed. And there's also a requirement actually to update it with the data. And so for any clinical trials you want to participate, you can go on there and you can see where they are in the recruitment process, what data they've been able to release. We're not required to release everything, by the way, no company really is, but it's the main things that you've put in already as far as your primary endpoint, secondary endpoints. You have to at least put as much information there to show once it's completed. And there's a timeline specific there. Phase 2 data can be talked about early just because it's not blinded. Phase 3 data is blinded, and blinded just means that neither the participants, the investigators at the site staff, or the sponsor, Aardvark, actually knows what's been dosed. So you see placebo and treatment arm. We have to wait until everyone has actually been enrolled and dosed and completed dosing. And then we stop the study in terms of when we collecting new data. All that data has been entered. Once that's done, and we've ensured that there's no new data that's going to be entered, we can look at the blind, we can unblind the study, but it's until that very last subject completes dosing and goes into open label extension. And so that's an important piece. So then we don't actually know the data until that happens. So that's for every single phase 3 trial. And at that point, we look at you probably have some like words in the back of your mind, like top line data. That top line data is based on when that data has been unblinded. And that's very, very important for a phase 3 trial because you do not want to introduce any bias by the investigators, by even the participants, or by the sponsor. And so it's the cleanest way to run a clinical trial. So that's why it takes a long time. Now, we're kind of newer in the space in terms of the execution of these trials. So you haven't seen as much data just because we haven't been around as long and we haven't had these phase 3 trials and then an open label withdrawal trial that you're referring to in terms of Soleno data. And that's just because we haven't been around as long, but we're doing the phase 3 now. And as soon as we have the unblind available, we're going to have top line data. You better believe we're going to publish that. Absolutely. We are so excited to show you that. So I wish we could do it tomorrow. But this is why we need this trial to continue and enroll. And it's because we truly believe in what we're doing and really, really excited, as you can tell.
Yeah, and one more thing, I just want to make sure I just address some of the other active questions as far as the diversification, for ethnicity, race, gender. Definitely high in mind. We have this really as an issue. We were trying to make sure we have representative patients. Maybe Monty, you can address that we'll quickly too.
Yeah, absolutely. And so one thing that we're also doing, and I'm actually hoping to do this in a month live, is really last year, I'd recently actually just joined Aardvark and we met with the BIPOC community. We had a breakfast. It was very influential for me starting out at this company to understand where the limitations are. So I really want to work with advocacy groups to really make sure that we address questions and participation specifically. Will we know, are we meeting the participation goals? I don't know if we'll know it in real time. So instead of kind of falling back on it and retrospectively looking, I want to be reactive or proactive, not reactive. And so hoping that by having this conversation next month and kind of looking at it, and then now reaching out to the sites as well and circling back and saying, okay, where can we support you to make sure that we have equity across the board for participation? And so I would love to hear that. And actually, if you want, you can reach out to me directly to say, okay, this is how, or through Dorothea, maybe that's the better path to say, okay, we would like to kind of understand how we're addressing this. This is a suggestion that you may have on how participation can be improved. And we will take that and we will run with it.
I'd also like to remind everyone who's listening this evening, whether you're listening to a recording or whether you're listening live, that Aardvark is going to be at our conference next month, and they will be there with a booth and bells and whistles. So any questions that you have or comments, they will also be there ready to answer and speak to members of our community. That being said, I do want to be cognizant of time. There's still so many questions going on in the Q&A, which is wonderful. There's one question that's come up a couple times in different forms that I'm hoping you all can address, and it's a question regarding the impact of an individual's diet, specifically a PWS diet with limited sugar, and how you see that playing a part in how this drug might work.
Yeah, so we don't know what we don't know. My intuition is that irrespective of the diet of the individual, this drug should be effective because, as I mentioned, we really believe that the driver of the overeating for Prader-Willi is really driven by hunger, not the appetite. Maybe some foods are more appetizing, so that's another. But on relative balance, taking care of the hunger would take care of the hyperphagia in the majority of circumstance in our view. So I don't think it matters, frankly, in terms of the outcome, as long as it's consistent throughout the course of the study. Just don't change any things in the middle of the study. But if everything else being equal, we think that's we'll be able to demonstrate the benefit. Same rationale. I just want to address several questions of the similar theme about concurrent medications and so forth. If you're stable on a medication with just some exclusions that are noted, then you should be able to get going on the study. There's actually some specific medications that we want to we think that it would even work in the context of most of these medications. We just want to clean the signal, because if you have two groups of relatively small number, if you just have an imbalance in folks on one arm on this drug, then how do you separate the signal strength based on the math? That's what we're trying to solve for. And also ViCAT, we think ultimately those drugs are not really competitive in terms of exclusion, right? But just we want to keep a clean signal. So for now, for this study, we don't want to have a simultaneous use. But if you started ViCAT and you have interest to be in the study, as long as you're stable on off ViCAT, there's a way to enroll as well too. So it's all written in the protocol. So we want to accommodate. It's not like a situation where you'll make someone permanently ineligible unless there's like some other explicit exclusion. As far as the enrollment, it's worldwide. There's sites in the U.S., EU, and Asia Pacific, but it's a competitive enrollment. We really are motivated to try to get this trial complete as much as possible. So if we are surprised and there's a lot of enrollment, we might be done earlier, but we're not saying that set number of patients in each country. We're just basically trying to open the study up. So if folks want to enroll, and we wouldn't necessarily stop at our minimum. We may over enroll, but we just want to get this completed because we have high confidence that we'll be able to show what we believe to be true, that our drug is going to be effective here.
So Tien, to clarify, competitive enrollment means first come, first serve, right?
Yeah, so any sites that are up and they have patients, they can enroll, and that's important. It just allows us to move quickly so we can get this to you as soon as possible.
Right.
And then one thing I want to clarify on this image that I'm looking at, open label actually starts week 12. So it's only for those that don't go into open label or choose not to move forward in open label that you would have that follow-up visit, and then you would be done at week 16. Anyone who goes into OLE actually starts week 12. So that would be your first day visit of OLE. You go right into it. There's no gap. I just want to be very clear. We do not want a gap because we want you to just have a continuous, easy accessibility to the OLE as well. I just saw that. I was looking, I was like, wait, that's incorrect when it comes to the visual. So you will go right into the OLE after week 12. There will be no 4-week assessment unless you do not go into OLE. And that would be your choice not to. And then in terms of a couple of more questions then, how long do you need to have your medication stable for? So that's 3 months. However, if you're on chronic medications that has changes that are needed, like growth hormone, within the standard of practice, that would pay what the physicians would normally do, you can change that based on that. But otherwise, really just stable as for 3 months. I think we answered the diet question. We answered the current medications question. So group home participation, that's a good question. So what we would like is for the time frame of the study is the same caregiver at the group home who has that requirement of 4 waking hours on average per week, per patient, per day, per patient, would be the one who actually goes to fill out those questionnaires. That's the requirement. We took that into account. That's why there's that flexibility on average, but it has to be that same person who actually is following the patient at the group home. I hope that clarifies that piece. Oh, the cost, we cannot comment on the cost yet. I think that hopefully we'll have more information, but first thing is getting the phase 3, and then we'll kind of look at them. We're obviously looking at that, but we can't make a comment on that yet. Hopefully we can soon. And then I'm just going through. So yes, there was one patient who withdrew from the phase 2 trial, and we actually showed this in the abstract as well. This was the only patient who withdrew. Every other patient that's ever enrolled in the clinical trial completed the full clinical trial, which is why we're so encouraged with the safety. However, this one patient did withdraw within 3 days of dosing. There's some safety concerns from previous medical history for this person who also actually had a previous drug response 2 years before to something else, but had a previous history of this. So they took the drug, and day 3 they withdrew because they had some response to it in terms of some hypersensitivity is what it's falling under. But again, there's a previous history. This is also something that we've disclosed publicly, and we are, again, even with that one, we're reassured that the rest of the 12 really had nothing similar to that, and we know that there's a previous medical history, but we paid attention, and we are continuing to pay attention to these specifically. And that's where we're also doing inflammatory biomarkers just to see, are we affecting it? We actually think we're decreasing it, but we'll see in the larger subset with the phase 3 trial. Enrollment process. I'm not sure what that is specifically to, but if it's just for the phase 3 enrollment process, then I would encourage you again to go either clinicaltrials.gov or to Hero for PWS, because that's our website for the phase 3 trial. It'll tell you which sites are up. It'll tell you kind of the timelines for most. It'll probably help you, guide you to a service that we have. We like to call it a concierge service, but that allows you to actually, in the US specifically, it is kind of a concierge service, which we really wanted to make it as easy as possible for you. So they actually walk you through kind of what's your local site, what's the best site for you, how can we get you there? And by the way, Aardvark is also making sure that you have full transportation, hotel, lodging, all of that provided. So it decreases the burden on you fully. And again, that was really intentional because we understand the burden that a clinical trial has. So that enrollment process is actually handled through this concierge service that we actually are using, that will allow you to move forward in the enrollment, whichever site is closest to you.
And Nancy, you have 3 sites that are currently open and enrolling. Can you just folks, if you're paying attention to our pulse, our direct emails, our blogs, our social media stuff, then you probably already know this. But there are currently 3 sites that are open, active, and enrolling patients right now. Muncie, if you could.
There are, and there's actually more. So what I would say is right now we have 3. I will say we're anticipating at least a few more by the end of the month. And so pay attention to where these are. There's Texas, there's Alabama, there's New York, Delaware that's coming up. I mean, there's a few, but it's spread out throughout the US. We're trying to get every region possible. But also keep in mind, if you are not close to a site, but you're close enough adjacent, we can actually provide transportation to those sites. So if you really want to participate, I would say connect through our website. That concierge service will be able to connect you to a site that you can actually be sent to. And so it's important to know where it is, only because if you can't make it to one of the ones, if it's too far away, we can figure out a way for your participation because we want to support it. If you really want to be a part of it, we will find a way. Any other questions left, Dorothea?
I don't think so. There's a few that are still kind of looming, but I think we've spoken to a lot of what's around. There's one that talks about statistics related to weight loss while on the HERO study. What happens after phase 3. I think that was also addressed and in regards to the open label extension possibility for folks involved in the trial. And I'm pretty sure that you dove into the exclusionary criteria pretty well. I think at this point everything has been answered, unless Tien or Muncie, you can think of anything else that.
One thing I say, but I don't want to leave anybody hanging. So we think that mechanistically, weight loss is context specific, but we actually think body composition improves, which is actually pretty notable, right? We think we're actually helping with lean body mass and decreasing body fat. So it's not just about the weight, and it's actually related to the missing CCK signal. As far as what's after phase 3, I think potentially the FDA never gives you absolute guidance, but if we really get a strong signal, it's potentially a pivotal trial, and then we think we're in position for NDA application, which would lead to an approval if everything were to go to plan. Sorry, we're missing something. I just want to make sure. I just don't want to have a feeling that I'm missing someone's question. I do want to. We have answers for everything. I didn't want to inadvertently leave anybody hanging.
No, I think we're okay. I think the last one was about exclusion criteria, which was already talked about. But I think we're in good shape. Folks, again, I'm just gonna say this one more time. If you are coming to conference in Arizona next month, Aardvark will be there and ready, willing, and able to answer any additional questions that you might have. Their team will be there. There's also going to be sitting on the clinical trial panel, so you can hear a little bit more about the trial. There will be additional sites open up, so I'm sure that that will be also part of conversation that we're having there. And we are extremely excited to have you all working in our space. So thank you.
Thank you. Thank you. And I will say that at least half our company is going to be there. And that's because we really truly care to be present and support everyone that's attending and be able to answer questions, but also just be there to support the community. Everyone here is passionate about what we do. We care about our patients. Patients are our North Star. And Dorothy, you've heard me they say this line over and over again, but I live by this, is that, a protocol is just words on paper, and it can't come alive without the participants and their families. And we're forever grateful for this community to support us as we try to do this.